Hi everyone, I'm Dr. Taseen. I'm a microbiologist. With close to 2 million cases of COVID-19 worldwide, a lot of people want to know about what viruses are. We can't see them, yet the entire world is in lockdown. Some people have gone as far as calling these agents the invisible enemy. A virus is an infectious agent or particle that consists of two essential components, which is the genomic material and the viral capsid. The genomic material can be either RNA or DNA, while the viral capsid is a protein component that encloses or protects the genomic material. The viral capsid can have two geometric configurations. It can either be a helical capsid or an icosahedral capsid. In order for a virus to replicate, it has to enter a cell because it does not have the required machinery to replicate. The process of replication consists of two stages, the eclipse period and the exponential growth. In the eclipse period, this is where the virus enters the cell and makes one virion or one virus particle. This can take up to anywhere from 8 to 20 hours. The exponential phase is where the virus goes from one virion and can make up to anywhere between 100 to 10,000 virions from one single cell. What are the important steps in the replication cycle of a virus? The first thing that must happen is that the virus has to attach to a receptor on the surface of the cell, and following that the virus will penetrate the cell, enter the cell, and the protein coating, the capsid, has to now uncoat so that the genetic material can be released, whether it's RNA or DNA in the cell. At this stage, the virus will use the host cell machinery to make copies of the genetic material. So you have copies of the DNA or RNA, and it will also make copies of the proteins so that the virus is now ready for the final stage, which is to assemble virus particles and release progeny or virions into the surroundings. When a virus enters a cell, there can be four outcomes. You can either have an abortive infection, where the virus enters the cell but is unable to replicate and successfully create progeny viruses, so the virus is destroyed. You could have a productive infection, where the virus goes in, replicates, makes many copies, and then can infect other cells, but the host cell is not killed. Or you could have a productive infection where the host cell is killed, so the virus goes in, replicates, makes many copies, and can now infect other cells and the host cell is killed. And lastly, you could have a latent infection where the virus goes into a dormant stage where it can either stay inside the host cell for long periods, or it can go in and integrate its DNA or its genomic material into the host cell genomic material, so now it becomes part of your body's cellular DNA. Thank you so much for watching, and stay tuned for more videos.